Cholesterol is a lipid molecule that aids in the maintenance of cell membrane structure. It's a building block for steroid hormones, bile acids, and vitamin D. The majority of our cholesterol is produced by us. However, some is obtained through our diet. Cholesterol production, commonly known as the Mevalonate Pathway, occurs in the smooth muscle of the body, the cell's endoplasmic reticulum. It begins with the enzyme joining two acetyl-CoA molecules together, acyltransferase acetyl-CoA. The outcome is acetoacetyl-CoA, a 4-carbon molecule, followed by a free CoA molecule. The enzyme HMG-CoA synthase then joins acetoacetyl-CoA and acetyl-CoA to generate a new compound. 3-hydroxyl-3-methylglutarol-CoA, or HMG-CoA, is a 6-carbon compound. After 3 acetyls and a free CoA molecule, HMG-CoA is reduced to mevalonate by an enzyme called HMG-CoA reductase, taking off a CoA-SH from a water molecule. The rate-limiting step in cholesterol production is the HMG-CoA reductase process. In other words, the overall rate of cholesterol production is determined by the rate of this process. It's like the slowest step in a factory's assembly line. A pair of proteins now control cholesterol production. SREBP, or sterol regulatory element binding protein, and 2 additional proteins known as SCAP and NSIG1 assume that cholesterol levels fall as a result of less cholesterol entering the cell from the food. In this case, NSIG1 is removed from SREBP. It's like removing a grenade pin. Cellular enzymes then break the SREBP SCAP complex. SREBP that has been cleaved floats into the nucleus and binds to the DNA sterol regulatory element. When it binds, it raises the expression of genes that code for HMG-CoA reductase, resulting in more HMG-CoA reductase. This increases the rate of endogenous cholesterol production. Once the 6-carbon mevalonate has been produced by HMG-CoA reductase, it subsequently travels through a series of enzyme-mediated changes before being used. It turns out cholesterol. To begin, adenosine triphosphate, or ATP, is used by the enzyme mevalonate 5-kinase. Mevalonate is phosphorylated to produce mevalonate 5-phosphate. Phosphomevalonate kinase then phosphorylates mevalonate 5-phosphate to produce mevalonate pyrophosphate using another ATP. Finally, mevalonate pyrophosphate decarboxylase removes a carboxyl group, resulting in isopentenyl pyrophosphate, a 5-carbon molecule. Geranyl transferase then condenses 3 of these isopentenyl pyrophosphate molecules to generate farnesyl pyrophosphate, a 15-carbon molecule. The enzyme squalene synthase then combines 2 molecules of farnesyl pyrophosphate to create squalene, a 30-carbon molecule. The oxidosqualene cyclase enzyme then transforms the linear squalene molecule into a ringed shape, a process known as cyclization. The outcome is lanosterol, our first sterol intermediate. There are then 19 processes of sequential modifications that transform lanosterol into 27 different compounds. Carbon 7D hydrocholesterol is converted to carbon 27 cholesterol. It's important to remember 7-D-hydrocholesterol now. That's because cholecalciferol is produced when a single photon of light strikes it in just the correct way. Vitamin D3 is necessary for the metabolism of calcium and phosphate. Now, the majority of cholesterol is produced and consumed by the liver, eventually resulting in bile acid. Bile acids are divided into two categories: chenodeoxycholic acids and cholic acids. Amino acids conjugate both of them. Taurocholic acid and taurochenodeoxycholic acid are formed when taurine is conjugated. Glycolic acid is converted to glycodeoxycholic acid by conjugation with glycine. After meals, these bile acids are retained in the gallbladder and released into the intestine to aid in fat digestion. Some bile acids are excreted in the feces from the gut. The majority, however, are reabsorbed by the colon and enter the portal circulation and return to the cells of the liver. Because cholesterol is insoluble in water, it must go through the bloodstream via lipoproteins. Which are part lipophilic, which loves fat, and part hydrophilic, which loves water. Apart from the liver, every cell can now produce cholesterol on its own. Cholesterol is used by cells in a variety of ways, including as a component of the cell membrane, where it serves a dual purpose. It squeezes in between phospholipid molecules at low temperatures, preventing them from packing too firmly together to maintain a more fluid membrane. Cholesterol binds phospholipid molecules together at high temperatures. Cholesterol makes the cell membrane fluid and durable regardless of the weather by minimizing the gap between them. Finally, the adrenal glands and gonads can use cholesterol to produce steroid hormones. To create corticosteroids, enzymes in the cortex of the adrenal glands tinker with cholesterol molecules. Cortisol and aldosterone, for example, play a part in our fight or flight response and blood pressure regulation. The gonads have a unique set of enzymes that process cholesterol in a unique way. Cholesterol is mostly used by the testes to produce testosterone, while cholesterol is primarily used by the ovaries to produce estradiol and progesterone. Now for a recap. The smooth endoplasmic reticulum of cells throughout the body produces cholesterol. The reduction of HMG-CoA to methylate by HMG-CoA reductase is the rate-limiting step in this process. Squalene, the final linear precursor in cholesterol production, is formed by combining the molecules. Squalene is cyclized to produce lanosterol, which is subsequently modified to produce cholesterol. Cholesterol is mostly found in bile acids, which aid in fat digestion, but it is also involved in cell membrane fluidity and steroid hormone synthesis. That's all for this one. Thanks for watching to the end. Don't forget to like and subscribe, and tap the notification bell so you'll know when we upload a new video. We'll see you in the next one.